The overall goal of this procedure is to measure thrashing frequencies of freely swimming microscopic organisms. This is accomplished by first preparing a sample of microscopic worms. Next, the optical setup is prepared. Then, the diffraction pattern introduced by the live organisms is recorded. Finally, the recorded data is analyzed to determine swimming frequencies. Ultimately, diffraction analysis is used to show changes in locomotion patterns of unconstrained worms.
The main advantage of this technique over existing methods like traditional microscopy is that swim patterns can be observed as the species moves freely in a three-dimensional space.
Though this method can provide insight into freely swimming C. elegans, it can also be applied to other transparent, small, and microscopic organisms exposed to varying conditions.
To set up C. elegans for video analysis, begin by using a flattened platinum wire pick to move 10 to 20 gravid adult nematodes to fresh NGM agar-filled Petri plates containing small, circular spots of E. coli. Allow the nematodes to lay eggs for 3 to 5 hours, and then remove the adults, thus establishing a small, developmentally synchronized culture of 50 to 150 nematodes. To grow the nematodes to early adulthood,